Our week 9 topic builds upon what we have previously talked about in week 8 relating to animals. And so for week 9, we're looking at zoo archaeology specifically. So I'd recommend that before you watch this video, before you get into the week 9 notes, and before you get into the subsistence lab, that you review the topic 8 or week 8 materials as well. So what are we going to do? Well, we're going to get more into zoo archaeology, focusing on the methods we use to analyze faunal assemblages. Okay, so for those of you who've taken biology, some of this in terms of terminology and classification of animals will be a little bit of review. Again, that's kind of background information. What I want to focus on, because what's critical for archaeological interpretations, for getting at, again, people in the archaeological record, we need to look at how we analyze faunal remains. So the way we're going to do this is by looking at how we can reconstruct animal diet, so not human diet, but what animals were eating through sexing animals, so doing sex estimations, aging remains, so age at death estimations, and then we'll talk about some other forms that we can quantify faunal assemblages to get a broader perspective at how animals were being used by people. Okay, so again, some terminology we're going to be talking about. Some of the key terminology I'll be using is specimen, which is when we have like an isolated bone, tooth, or shell. It can be a fragment or it can be complete. Typically, when we talk about elements, they are complete. Fragments, of course, are not complete. They're incomplete or broken. Of course, we're going to talk a little bit about taphonomy too, and that's really critical because one of the things we want to know is if we have complete and incomplete elements, what's going on? What are the processes, both the cultural and natural transforms, right, that have shaped the faunal remains as we recover them in an archaeological site. When we talk about samples, this is when we are selecting as an archaeologist from a broader grouping of specimens, or from within an assemblage, or from within a site to better understand what's going on. We can talk about collections, where we have samples from a single time period, and assemblage, which is when we have multiple occupations resembling different time periods at a site. Different types of bones. Because of some of the limitations we're dealing with being online content delivery right now, you won't be interacting with any physical specimens, with any faunal specimens or elements or fragments in the laboratory environment, but it's just useful to be familiar broadly with the different kinds of bones that are available in a skeleton. So we're not going to force you or expect you to do any skeletal identification. Instead, we're going to focus on how you may be able to do some basic analyses on specimens. But here, again, is just a generic template of a mammal, giving you some of the names of some of the key bones that are present. Okay, and this is just kind of a brief review in terms of how we classify animals, and including ourselves, right? Using binomial nomenclature, using taxonomy, right? So this is a broad view of the system. Personally, I really like this figure, but I also don't like it in that, to me, it seems backwards, because I like to start with domain at the top, working down towards a subspecies, but that's my paleoanthropological training. But anyways, so this is an example showing how we get from the highest taxonomic rank of domain all the way down to a dog or a subspecies. So some of the big groupings we deal with, and I'll just mention that this is kind of what's a little bit different sometimes between zoo archaeology and zoology or biology in general, is that while we will report in our formal writing the genus and species and subspecies if known, we'll use the terminology that's appropriate for taxonomy. We still typically refer to animals by either their kind of larger groupings and by their commonly known names. And this is for a number of different reasons. We can make our evidence, our discussion, our interpretations, our analyses a lot more accessible to people if we use commonly known names. This can be important when we're working within and with communities in terms of the names that we do use for animals. It can signify the social and cultural significance of these animals to the people who still continue today to interact and utilize these resources. So there's a number of different reasons why we do this as archaeologists. And I should also note at this point that if you're really interested in archaeology, we do have a 300 level course that is dedicated to zoo archaeology, which is Anthropology 375, where you actually get to learn more about all of the how we classify faunal assemblages. You learn how to do that yourself and then also do analyses as well. So broadly speaking, we can talk about some differences between invertebrates and vertebrates. So two critical invertebrate groups are the mollusks and arthropodas. So mollusks, we've talked a little bit about how some of them are, especially bivalves, are really critical in terms of establishing things like seasonality. They're also an important food resource. Arthropoda were very interesting archaeologists because they are really useful for us for reconstructing the environment, right? Because remember that invertebrates represent a large part of our microfauna, and they're very sensitive and critical components of understanding things like seasonal change, like environmental change, like climate change more broadly. So both on regional and larger scale processes. In terms of vertebrates, of course, we have the major groupings here: fish, amphibians, reptiles, birds, and mammals. I mentioned again in the week 8 materials that there does tend to be almost a bias towards mammals and vertebrates in general, just because they are larger bones. Because of mammal bones and their structure, their bone structure, they tend to preserve a bit better. As you might be able to imagine that birds, because they have light hollow bones, they're a lot softer. They don't preserve as well in many environments. Fish have tons and tons of bones, but again, depending on the preservation environment, they're not as resilient as well. So we have different kinds of information based on, and different kind of frequencies of different kinds of faunal remains, often relating to the bias that's inherent in the archaeological record because of preservation. So how do we go about analyzing faunal specimens, which is what I want to focus on? Well, it's a multi-step process, right? So we begin with what element is present. We talk about, is it complete or is it incomplete? If it's a fragment, we talk about the proportion, and we have directional anatomical terminology we talk about, such as distal or proximal, medial, etc. We describe any anatomical traits or attributes that we find that are assisting us with the identification of the element, right? So is it a long bone? Is it a short bone? Is it cranial? Is it part of the axial skeleton or appendicular skeleton? So we go through this process. And then once we've identified the element itself or the element fragment, then hopefully we can attribute it to a taxonomic category. And as I mentioned here, we're generally conservative. And again, this is because we may not know, and we also have to acknowledge that some of the things we talked about in the week 8 materials, that there's also significant differences that can be between long-term domesticated species, but animals at the point of domestication or in the process of being domesticated. Sometimes that line between what is a domesticate and what is a wild variant can be difficult. Subspecies sometimes can be difficult to determine using just macroscopic analysis of faunal remains. So we tend to be conservative, and so that's why talking about, oh, this is a bear or this is a dog or a cat, this is why we will often use those broad categories versus focusing on getting down to the species level. Then we can start to look at other morphological features to get into other specifics about that individual that can then tell us about, again, the particular environment it was coming from, and potentially why it was being exploited by humans, how it ended up in an archaeological site. So the key way that we do all of these things is not only by studying ecological works, but by using reference collections and photographic atlases, and increasingly online 3D models as well that will assist us with our identifications and analyses. Then once we've done all that, then we can get into quantitative analysis, and I'll talk about some of those towards the end of this. So for dietary analysis, and I'm giving you a very introductory, basic introduction to these methods and techniques. So again, if you're very interested in this, take 375 where you really get in depth with this. But for example, if we want to reconstruct the diet, using faunal specimens, we'll look at dentition, because different kinds of teeth and different shapes of teeth, or the different surface morphologies of teeth, tell us about the different kinds of food they're using. So for here, I have some examples of what teeth look like in mammals that eat fruit versus leaves versus gums versus insects. And so most of these are actually looking at primate teeth. The larger picture on the right-hand side here is showing you the 4 major types of teeth that are found in mammals. So incisors, canines, premolars, and molars, but showing you how they vary based in the mouth of a carnivore versus the mouth of a herbivore. Because you can imagine, if we think of teeth as being tools, that you need different tools to cut, pierce, slice meat than we need for grinding up and processing leafy materials and other organic materials. Okay. We can also look at microwear. Of course, the kinds of things we eat impact our teeth themselves, and they can leave behind wear marks on the surface of our teeth, including even just intensive wear caused by chewing harder materials or more intensive use to break down and process materials, right? So that can be a critical means we can use to reconstruct diet. Here's some images looking at an early hominin called Oreopithecus and comparing it with Oranopithecus. And what you can see is that there's different kinds of striations that are left behind on the teeth based on what they ate. So with the image on the left here, we can see this deep pitting and grooves caused by consumption of harder materials like nuts or tubers. Whereas on the right-hand side, you'll see that it's very scratched on the surface, but there's not these kind of pits that are caused, and so it's been suggested that they're eating kind of leaves or other soft objects instead. Okay, also to get at diet, we can use the same means that we use to get at human diet, which is stable isotope analysis. And so this is an example of what the nitrogen signature looks like in various different kinds of organisms in terrestrial versus in marine ecosystems. And you can see that Ambrose included not just carnivores and herbivores, animals, but also looking at plants as well, and nitrogen fixers as well. And so you can see that there are different proportions of nitrogen 15 in carnivores and herbivores, so higher based than other organisms in terrestrial environments. And then in marine ecosystems, you can see they broke it up a little bit differently between, things like microorganisms like algae, all the way up to mammals and fish. And then I have a further description below in the notes explaining what we're seeing. Important here, note that some of the conclusions of the study talked about how marine environments tend to have overall higher nitrogen 15 values than terrestrial ecosystems. So even just the overall value that's encountered might tell us something about the ecosystem that the animal was living in. In terms of sex estimation, so we can look at size differences. So many species demonstrate sexual dimorphism, so a significant difference in the size and coloring of males versus females. So you can see mandrill baboons, the brightly colored fur and skin around the face in a male, and the overall large size versus in a female. Of course, peafowl, peacocks and peahens, we have the brightly colored plumage of our male versus the browns and subtle colors of the female that allow her actually to hide more effectively in terms of protecting her eggs. Praying mantises, we have sexual dimorphism in terms of size. Birds, lots of examples, right? Ring doves and jacanas also have pronounced sexual dimorphism as well. We can look at pelvis shape, particularly in mammals. Pelvises in females tend to be much larger and wider than in males to facilitate pregnancy, labor, and delivery of the fetus, of the infant. Baculum, this is a bone that is absent in human biological males, but it is found in many other mammals. It's basically a penis bone. We can have spurs present in many males. Antlers might be absent in females, present in males, or differences in size, again, relating to sex. And then also a medullary bone can also vary based on sex as well. In terms of age, we can determine this on a number of different characteristics. So degree of suture closure on bones. So when we are born, we have bones that are small and are basically incomplete. and they basically grow together. These various fragments of bones grow together, what are called epiphyses and diaphyses, or shafts and ends, grow together as we age, allowing us to have then an adult-sized skeleton. So the degree of closure may tell us how long in this process of growth an individual is, therefore something of their age. Tooth eruption aware, I have a slide about this, is really critical. We talked about growth rings in shell relating to seasonality, but of course this can also tell us about the age of that individual. And it's not just shell, but we can also look at growth rings in teeth and horns and claws and scales as well. And overall size is usually a good indication. Generally speaking, younger individuals are smaller than older individuals in many animals. So this is an example of how we can look at that example of epiphyseal fusion or growth in bones. So this is showing you where we have the long bone of a tibia of a young cow, and you can see that the ends have not yet fused on. So this is what the ends look like when they were separated from the shaft. And the idea is that as the individual grows, this cow were to grow, these joints essentially would fuse together, and the age at which this occurs is different based on what mammal we are dealing with. And so the approximate ages at months are provided here for various bones that are present in these various bodies. And so we can use these known growth rates and this the age at which epiphyseal fusion occurs, again, in known individuals, to estimate that for remains we find in archaeological sites. Here's an example of how we can use teeth. So you might remember yourself that as you were aging, you had teeth that fell out, and then your adult teeth, so those are called deciduous or baby teeth that fell out, and then you had adult teeth that grew in. The rate at which this occurs, again, can be observed, and what we see that there is these standard ages at which this period of eruption, loss, and then eruption and growth, and then wear occurs. And so we can see this in mammals as well, and so this is for a deer. And so you can estimate based on which teeth are present and the approximate wear of those teeth to get an estimation of age for a deer mandible or for deer teeth. Okay. So those are some basic forms of analysis we can use. And then after we do that, we can then zoom out from the assemblage and start, or from the element, and start to look at an assemblage level. And then we can start to apply different kind of basic forms of statistics or statistical analyses to tell us something, and really this is not so much statistics, but quantifiable forms of analysis to look at the data we have accumulated. And the two major ones that we use in zooarchaeology are called NSPA and MNI. So NSPA is the number of identified specimens. So this is simply a measurement of the number of identified bones we have relative to the total number of bones in the assemblage or in the sample or whatever. So to calculate NISP, it's fairly easy. We just sum up for our particular species or our particular taxa. So in this case, it's bison, the number of fragments we have and whole bones that we have. And so in this case, luckily, we have all elements that are complete. We don't have any fragments. So we have one skull, and we add that to the 3 right tibia or shin bone, to the 2 left femora or femur or upper leg bone for our bison. And so our NISP for this individual is 1 + 3 + 2, which is 6. Yay. So that's a fairly easy thing to calculate. We want you to do this in our subsistence lab. So please refer back to these notes as needed. The other thing we do is MNI. And so number of identified specimens simply tells us that is how many bones or elements in our assemblage were we able to identify, right? But that doesn't possibly tell us how many animals are represented for each taxa in the assemblage, right? It only tells us simply what we were able to identify versus what we weren't able to. And so MNI allows us to get at the minimum number of individuals represented by an assemblage. And this is because if we go back, sorry, I'm just going to scroll all the way back to this diagram of a general skeleton. So this is of a pig, but it applies to something like bison that we're using for our example as well, is that we have things like 2 front limbs and 2 back limbs, right? We have tons of vertebrae, tons of ribs, right? Tons of tailbones, right? So what does it mean when we find, a skull, But then we find 4 different leg bones. You know, how many individuals are possibly represented by the bones we have in our assemblage? And that's what an M&I allows us to figure out, right? Because if we have a skull and every organism only has one skull, it's easy. But when it comes to other bones that are present in the skeleton, this can become much more challenging. And the reason why we want to know the minimum number of individuals that could be represented for particular taxa within an assemblage is that it allows us to make different kinds of interpretations based on, you know, how many of a particular species we're being selected for, right? You can imagine fish have tons and tons of vertebrae in their backbone. So just because there's tons of vertebrae found in a site doesn't mean that there's actually a lot of fish that were captured and being used by people, right? Plus, we have to then think about relative sizes of animals. So we have a whole bunch of different metrics we can then apply once we get some of these basic quantifiable steps taken care of. Okay, so enough about that. Let's talk about how do we figure out M&I. Well, the formula for M&I is we take the number of each identified element for that taxa and we divide it by the frequency of each element in an individual. So skulls, figuring out the M&I of an individual based on skulls is pretty easy because we know for mammals that you should only have one skull, right? So if you have one bison skull, as we do in our example here, we divide it by 1. That means that the M&I of bison is 1. However, we have more than one bison remain in our assemblage, and so we have to compare how many skulls we have to how many leg bones we have versus how many vertebrae, et cetera, et cetera. Okay, so based on the skull alone, our M&I is currently 1, but now we have to look at the tibia. So there are 3 right tibia in our assemblage, but there are 2 right tibia there's only 1 right tibia that's present in each individual. Sorry, the reason why I was saying 2 is that there's actually 2 tibia in each bison. There is a left one and a right one. So in this case, the siding has become important. So if there is only 1 right tibia in an individual, but we have 3 right tibia in our assemblage, 3 divided by 1 means that our M&I of bison is now 3. So even though we only have 1 skull, in order to get 3 right legs, we have to have 3 bison as a minimum, right? There might be more, but this is why it's called M&I. And then we do the same thing for the left femora, right? Or the left femur, another leg bone. So there are 2 left femora in our assemblage, but there's only 1 left femora in each individual. And so this is saying that the M&I is 2. So now that we've calculated the M&I for each element, we can look at our overall assemblage and we realize that the highest number that we have is 3. So that means that our M&I is 3. Okay, and so I go through each of these steps as follows. So again, step 1, we're going to identify all the elements. Then we are going to identify the number of elements in a single individual. So again, 1 skull per bison, 1 right tibia per bison, 1 left femur per bison. Then we're going to calculate the M&I for all assemblages, rounding up to the nearest whole number. So this is what I did off the top of my head, right? So if we look at the skulls, we have an M&I of 1, the right tibiae, we have 3, and femora, we have 2. And so the highest value of elements is our M&I for our assemblage. For our M&I for bison and assemblage is 3. So our NISP or our NISPA is 6, but our M&I is 3. Oops, wrong way. Okay. The next thing we can do then is moving away from quantitative analysis is to go into some qualitative analysis. And we can just start to think about the kinds of faunal deposits we're finding, right? So why would we have an M&I of 3 bison at a site? Well, potentially because it was a kill, a collection, or a processing site. And the M&I of bison may vary depending on those different types of sites. But of course, killing, collecting, and processing animals isn't the only way that we engage with animals. Sometimes we might find them in residential refuge. Think if you have any animals for your dinner, you may discard their bones in your garbage, right? So middens is where we might find it. But if your beloved pet were to pass away, you probably wouldn't just throw it in the garbage, although I've never forgiven my mom for throwing away my little budgie when it passed away when I was in kindergarten, because I wanted to intentionally bury it. Why? Because my budgie was not garbage. My budgie Hawkeye was my beloved pet, and I wanted to intentionally bury him in the backyard. So we have to think about this. The context in which we find faunal deposits tells us something about their purpose and what they were being utilized for. How can we sometimes figure this out if the context is unclear? Well, again, this takes us back to taphonomy. And when we talk about taphonomy and faunal remains, really what we're talking about are the specific processes that alter the appearance of animal bones. And there can be a number of reasons for this, right? Cultural processes from killing, skinning, butchering, cooking, preserving specimens. Animals can impact other animals themselves, gnawing on the ends of bones, digesting the bones through the system, trampling the remains in the environment. And of course, the environment itself can impact it. All those different things like climate, temperature, whether the soil is anaerobic, aerobic, so oxygen rich or not, the soil pH, all of these different things can impact what the bone looks like, including things like root etching. So this is a picture of some root etching on some bones, and we can compare it with gnawing on a bone versus a bone that has been burned. Okay. So I love this model because what it shows is that it's never really ever one thing when we deal with faunal bones, that often there's a whole bunch of different things that occur in this taphonomic process. So we begin with our living community, right? And this is not what we're dealing with as archaeologists. What we're dealing with is the fossil assemblage. But in between the living community and the fossil assemblage are all these different processes that then impact what is actually visible in the archaeological collection. Of course, even the fossil assemblage itself may not be what we study as zooarchaeologists, and that then impacts how our interpretations or our reconstruction of that animal environment. One of the other things that's really interesting is we have to think about, again, how we have interacted with animals in the past. And there's been some lovely work that was done of early hominins that demonstrated that Australopiths in particular in South Africa might not have been hunters, but they most certainly were hunted. And so this was the work of C.K. Brain, who saw that several Australopith skeletons had these puncture marks, these alterations in their skulls. And what he was able to see was that they actually aligned with the canines or the teeth of these cats that were contemporaneous with them. And so what he argued was that, yeah, these Australopiths were actually being consumed and eaten by these giant cats. So carnivores do leave a lot of damage to bones. So this can be from rodent gnaw damage, as represented here, to step fractures, to various forms of spiral fractions. Usually you can see that these are caused on the ends of bones because carnivores tend to consume the shafts, or the ends, leaving behind the shafts. They also consume the ends to get at the marrow that's located in the shafts, and so that's why they'll gnaw on the bones to get out all that really rich, fatty goodness that is located in the marrow. So sometimes the frequency of shafts in assemblage can suggest if it's natural or cultural, so that's why it's important to look at whole versus complete elements. A lot of complete elements being left behind, particularly in the hoofs and the feet, parts of the animal that we don't necessarily consume, would be different than what we'd see in a carnivore assemblage where there's lots of gnawing and breaking up of teeth or of foot bones and stuff like that. Butchering will also leave different kinds of marks on bones. Gnawing damage tends to be very shallow, tends to be very rounded from the tooth marks, whereas cut marks on bones are very deep and V-shape in profile. Plus, they tend to be parallel, right? Because when you're cutting on bone, you're taking your tool and you're going in the same direction over and over. Whereas just simply gnawing on bones, you're going to be in all these different kinds of directions. We can also think about where we might butcher an animal, right? That cut marks are going to be left on bones with kind of quote unquote better cuts of meat. So we're going to see where muscles are attached. We're going to see where limbs are being dismembered, again, where joints are being broken apart. And all of these things can tell us different kinds of information about where the cut marks are and the frequency of cut marks, and then what's been left behind in the archaeological assemblage versus or in that particular assemblage, that our particular site versus which was transported away with people, tell us things about things like trade and consumption, about differences in cultural or ethnic groups, but also status as well, right? Different cuts of meat will end up with different status to individuals. So how do we figure out all of these different things? Well, through ethnoarchaeology. So you'll recall in week 8, we talked about archaeobotany or ethnobotany, where we look at contemporary practices, and then we use that to interpret practices in the past. So ethnoarchaeology has been really useful for figuring out how and why we butcher carcasses, the marks we leave behind, the kinds of tools we use, how we open mollusks and harvest them, for example, how we cook food and then discard refuse from these different practices, from these different stages. Okay, so that's what I had this week. Again, we'll revisit this topic a little bit more when we do the subsistence lab, and we're going to compare some of these methods that we use for analyzing faunal remains with those that we use for analyzing human remains in our week 10 topic, which is bioarchaeology.